Okay. All right. Well, then we're going to go on and we're going to talk. Well, actually, first off, so I gave an epic last night. Some of you were at it. I brought this in because this is what I wanted to bring in last night. I started it like 3 o'clock yesterday, and I thought it would get going faster, and it didn't. And there's sometimes variable, but this is the sort of this is cool because you get to really see the yeast. These are making a lot of carbon dioxide fast right now. Okay, so these guys are probably busy making alcohol. In fact, I think I can smell it um, right now because they've made enough carbon dioxide. They've displaced all the oxygen. This is all carbon dioxide in here. Um, so anyway, I think it's cool when it gets going like that. It's not something if you end up brewing. This happens with rapid rise yeast. It doesn't happen with like if you use a good yeast. It doesn't go. It doesn't do that. It doesn't burn through it right at the beginning so fast. Anyway, so that's an example of what that looks like. And we're going to move on and talk about membranes and getting things across membranes. So I was talking about this concept last time, and I want to talk about it a little bit more, and then we'll look at some examples of membrane transport proteins. So transport proteins are going to move, or let's say membranes can have an electrochemical gradient across them. So membranes can have an electrochemical gradient. And when I say they have an electrochemical gradient, there's two parts to this, right? We have electrical and we have chemical. And I talked a little bit about this last time. What was going on is, so this is an example of a chemical gradient here. We don't have any charges going on. We've got some molecules. There's more of them on one side than the other. And so what will happen is over time, they will move from where these orange dots will move from where they're more concentrated to where they're less concentrated. But if I wanted to move and when they do this, they will release energy. Energy will be released in this process. If I wanted to take one of these and I wanted to push it this way, I would have to put energy in for that to happen. Okay, and then once they're at equilibrium, there is no chemical gradient here. No energy is expended moving this direction or this direction. All right, so this is an example of a chemical gradient. All right, and then down here we have an example of an electrical gradient. So we would have a potential. You'd say we have a potential across this membrane. We have more positives on this side than we have on this side. So if we want to move negatives this way towards the positives, that's down an electrical gradient. So if that's all that's going on, energy would be released there. We'd also be moving those negatives up a chemical gradient, right? Because there's one negative and a bunch of negatives over here. But just if we think about just the chemical gradient part or the electrical part here, so our negatives are moving down an electrical gradient. Our positives in this direction are moving down a electrical gradient. And then if I wanted to push this negative, I'd have to push it up an electrical gradient to get it over here. If I let these things just go ahead, they're going to distribute themselves evenly by charge. Okay, and if I have a mixture of charges and concentrations, they're going to distribute themselves actually by a mixture, but you're not going to get a perfect mix of each depending on what's going on, okay? Depending on how many of each kind of charge you have. So what we have then is, so this is an example of an electrical gradient. And so we can put the two together with an equation that I'm going to show you, and then I'm going to ask a question. But so we can talk about free energy of things, right? So if we think about the free energy of a process, the actual free energy is going to be equal to the standard free energy plus a term RT ln of Q, right? Where Q is the concentrations of things. This is when we don't have that electrical component. Okay, this isn't right now made for membranes, essentially. But a standard free energy, what are the concentrations of reactants and products? And what I want you to think about here is let's think about this one, or think about it for this. But you've got, what are the concentrations of reactants and products where this is perhaps reactants and this is perhaps products at standard conditions? So let's pretend we've got a membrane here, and let's pretend we're in a cell, and we have inside the cell and we have outside the cell, and let's be talking about glucose. So glucose is uncharged, so we'll just have some glucose molecules. What are the concentrations at standard conditions? 1 molar, right? 1 molar everything. So there's 1 molar products if we consider out to be products, 1 molar reactants if we consider reactants, right? So if the concentrations are the same and the reactants and the products are the same, we're talking about something crossing a membrane. So it's glucose and it's glucose, and their concentrations are the same. What's the free energy change for moving across the membrane at standard conditions? 0. Did you say 0? Good. So it's 0 because essentially I have the same thing inside and outside, and I have them at the same concentrations. So that goes away. So what happens is we could think about the free energy change for crossing a membrane, and it's going to be equal to this other term. I got something wrong here because I have to, this ends up being negative, right? Help me out. I think is this just negative to be, it's not negative to begin, is this negative to begin with? Oh no, I know what it is. Sorry. Usually when we set this to negative, 0, right? We say when we're at equilibrium. That's what I'm confusing. Okay, so we don't set it to negative, but we have this term then that corrects for the actual concentrations of our reactants and our products. And here our reactants and products are inside, it's inside or it's outside, whatever substance we're looking at. And so I'm just going to define that as out versus in. So the concentration of out versus in. All right, plus we have another term. Okay, plus we have a term that, so this would be our chemical gradient. Okay, and this over here, then we're going to have a term, we have the charge, all right, +1, +2, -1, -2, okay, times Faraday's constant, which is 9600 something or others, okay, times the voltage. So this is the potential across the membrane in volts. Faraday's constant. Okay, and then this is the charge on molecule in question. The one that you're thinking about, is it in or out? Molecule or ion. So when I say ion, I mean it's going to, the molecule itself is going to be an ion, or we're not, the term is not going to count. So, but that's the charge with the sign. So this so here's what my question is going to be before I go on. If I take this equation or something like it, Nate, you've studied hard for the DAT. Was it on the DAT at all? Or in your study materials? Do you remember anything about that? Okay. Um, Bridget, you're studying for the MCAT. Has it been in your study materials at all?
Possibly. We, like, pieces of it. I just started this section in the physics book, though.
All right, we'll get back to, oh, well, in the physics book. All right, we'll get back to me on that. I'm going to cover it conceptually unless Bridget comes and we're not going to use it unless she comes and tells me that it's on the MCAT. Okay. Otherwise, I just want to talk about conceptually. I could give you a problem where it would be really obvious. There'd be no math involved, but I could fix the concentrations and or the ions such that I'd say, is this going to be spontaneous or non-spontaneous for them to move there? Do I have to put in energy to make them move? Does that make sense? So if I took the example right here, let's say I make this membrane semi-permeable, okay? And there's lots of semi-permeable membranes. The inner membrane of the mitochondria will not let protons pass, okay? So if this was a mitochondrial membrane, let's say, I could have a whole mess of protons out here, which would give us positive charge. So with this situation right here, if I take one of these negative charges and I move it across the membrane, is that going to release energy or is it going to take energy to move it across the membrane? How many people think it's going to release energy? Okay, one person, two people maybe. How many people think it's going to take energy? Okay, it's going to release energy because I chose this side because I figured there was about the same number of minuses. In fact, they're exactly equal, right? So there's no concentration, whatever this minus thing is, there's no concentration gradient there. And so these minuses are sitting here, and there's 1, 2, 3, 4 pluses over here. The volume looks about the same, right? And over here, there's 1, 2, 3, 4, 5, 6, 7, 8, 9, 10 pluses. So there's a gradient here that's going to make the minuses want to move that way. It's going to help pull them across, and energy will be released when they do that. Does that make sense? Why was it not clear? You guys had it the opposite. Most of you had it the opposite who talked about it. Anybody want to comment on why that wasn't clear or tell me what you were thinking? Or is it perfectly clear now? And is it clear now? Okay, great. I just want to make sure people have that. So that's the sort of question I'll ask you. Okay. Barring Bridget telling me that I should work the equation in greater detail, and I figure she probably won't even if she sees it on the MCAT at this point. But let's move on. And what I want to do then is give you some examples of some of these transporters. Actually, before we do that, let's talk about, we didn't talk about energy use by transporters. So we can classify these into two broad categories, and then we can break one of the categories down a little bit. So we can talk about something that doesn't take energy. So if a transporter does not take energy to move something across the membrane, whatever that something is must be moving up or down an electrochemical gradient. Down an electrochemical gradient. And it will not be able to push something up an electrochemical gradient. Okay? So passive, we talk about this as passive transporters. I'm going to say passive transport because that's how it usually gets referred to. No energy is used in moving solutes across the membrane. Okay, so no energy is used there, moving those solutes across the membrane, but also you can't put, solutes must move down an electrochemical gradient. Okay, for any kind of bulk unit. movement, right? If it's a passive transporter, you could have solutes moving back and forth all the time, right, at equilibrium. And even not at equilibrium, some would go uphill just randomly, but more would go downhill until you hit equilibrium. All right, and so at least in bulk, that's what I mean by the in bulk then, right? I just want to have people remember this is a dynamic system. In bulk, solutes cannot move up the electrochemical gradient. And they can't do that because it would take energy to do so, and a passive transporter has no way to use energy. Okay? Questions on that? All right, so then let's talk about active transport. All right, so energy is used to move solutes across the membrane. Okay, and energy is used, and so since energy is used, solutes can be pushed up an electrochemical gradient. Does that make sense? Okay. So if we think about evolution being efficient, right, using the least energy possible, if you're generally moving something, if something is going to be moving down an electrochemical gradient, which of these would be used? You're going to use passive. You're going to use less energy if possible. Yeah.
You said can be pushed up.
Yes.
Implying it can also be pushed down.
You could push something down. You could imagine something where it just always had to go.
But it's like rare.
It's less efficient.
Okay.
So you wouldn't see that from the perspective of evolution. And so if I ask you about that, and I do have a question I ask about that, it's going to have a statement about efficiency in evolution. Considering that evolution is efficient, what do you think this would do? Would it be active? Would it be passive? Which one would it be? So we have two types of active transport. We have primary active transport. Okay, and we have secondary active transport. Primary active transport, an energy molecule is directly consumed to push solutes across the membrane. All right, in secondary active, so that'd be like hydrolysis of ATP. It doesn't have to be. There are times that, like other types of molecules are used. Certainly in, various microbial systems, they'll use all sorts of things depending on their environment. In secondary active transport, okay, a gradient that has been produced by active transport, essentially, but a electrochemical gradient is used to move something across the membrane. Now, this can be a little bit confusing. What I'm talking about is when we talked last lecture, we ended up talking about uniport symport and antiport. Okay? So when we talk about secondary active transport, what's going on is perhaps something's been pushed out, like S2, and then it comes back in through active transport, and S2 comes back in with S1. So S1 is moving up an electrochemical gradient, and S2 is moving down an electrochemical gradient, and it is being used to help power bringing S1 in. Does that make sense? And then antiport would be the same thing, except we're going different directions. All right, so your mitochondria, what happens in your mitochondria for the electron transport chain is that protons are pushed out, and then they're let back in to make ATP. That is not secondary active transport, pushing them out so they can come back in by themselves. Okay, they are being used to do work. And there is an example, one of the protons actually comes back in to help bring a phosphate in with it, and that is secondary active transport. Is secondary active transport, people often get confused on that. Does that seem clear? Okay. So let's go back and let's look at a couple of examples of transporters, and you need to know these two specifically. This is the one I referred to last lecture, but didn't really go into detail. I'm going to show you the whole thing. So we got a cycle here. This is the sodium-potassium ATPase. And what it does is, through a cycle, what it does is it's going to push 3 sodiums out and 2 sodiums in. And this is pretty cool the way it works. And it hydrolyzes ATP to do that. So what would this be for an example of transport when we think about energy? Oh, I'm sorry. Did I say sodium twice? Potassium is coming in. So cells generally have a high concentration of potassium inside and low out, and then high concentration of sodium outside and low in. And that's certainly the case for your neurons, and they use that energy to transport electrical signals. They use it for transporting an action potential. But lots and lots of cells use this gradient. So this is the transporter I talked about that is consuming 25% of your energy right now. Okay, maybe a little less of mine because I'm standing and moving a little bit, but probably not much different. So it's 25% of your resting energy just goes into doing this. Okay. So we'll start here. You could start anywhere because it's a cycle, but let's start. What happens is this transporter, at least for this cartoon view, can open to the outside and it can open to the inside. All right, and depending on whether it's open to the outside or the inside, it has active sites that can, or it has sites that can bind potassium. These are potassium binding sites, or sodium. These are sodium binding sites. And depending on which side it's open to, it has a different affinity for those. So in this case, the transporter right here is open to the inside. And when it's open to the inside, it's going to have a high affinity for sodium right now. Okay, and while it's open to the inside, it's going to have a low affinity for potassium. So not only is this thing doing this wagging back and forth, opening to the inside and the outside, but it's changing its shape and affinity for these two ions when it does that as well. And that's how we're going to manage to push sodiums out and push potassiums in. And this moving back and forth then is powered by ATP. So here we hydrolyze ATP, we phosphorylate the enzyme. So what do we know about a phosphate? Lots of negative charges, right? So we have lots of negative charges, so that's helping to drive this shape change. Probably have 2 negative charges associated with that. And now our transporter is open to the outside. And it's going to have a low, in this conformation, it's going to have a low affinity for sodium. And when we go ahead and we scoot down here, you're going to see this, so the sodiums are going to be released, and the only place they can go is outside because of the way this is open. And at the same time, now we have a high affinity for potassium over here.
So the presence or absence of the phosphate affects the affinity,
It affects the shape, which affects the affinity, as well as whether it's open to the inside or the outside. Okay. So we're in this case. So here we see the results of that. I'm not going to write it again until we change shape again here. But so now we load up, we release the sodiums, and we then pick up potassiums from the outside, right? We're open to the outside here still. We pick up potassiums from the outside, and then the phosphate will get hydrolyzed off. The enzyme has some sort of phosphatase activity there. It's going to hydrolyze that off, and now it's going to open back up to the inside again. And we're back to where we have, this is a low affinity for potassium. Okay, and out here on the outside, we're now going to have a high affinity for sodium. Does that make sense how that works? So now we're ready to grab some sodiums again from the inside and then open up and pop them out on the outside. So this thing goes back and forth like that, and that is powered by ATP. And at the molecular level, or as close as this cartoon is, we've got this phosphate that attaches, and when it's attached, we get one conformation, and after it's hydrolyzed off, we get the other conformation. Questions on how that works? Okay, so we push, so it's 25% of your resting energy, right? A lot of this is going on. So you figure it would have a big effect. How many sodiums do we push out?
2.
3. How many potassiums did we push in? 2. What are the charges on those? So what's the net charge on the inside of a cell? Of your cells, of most of your cells, let's say. Well, not -1, but minus, right? Because they're continually doing this, right? So your cells are more negative on the inside than they are on the outside because of this. And that's kind of the point of, why this happens. So like I said, for nerve cells, I'm not going to give you the example here, but nerve cells are going to use that. They're going to let the ions back in to generate an action potential and communicate it, or not communicate it, but have an action potential run down like an axon. And then other cells will use the sodiums that are outside. They'll let the sodiums in with something else to help power bringing it in, secondary active transport. This is active. This is primary active transport. Questions before I go on? All right, let's talk about the GLUT transporter. So this gets used in all sorts of cells. Is this one named? It is not named. So there are a number of different types of GLUT transporters. Not in this class, but so much next semester, we'll talk about this in the context of the liver, because the liver's got this important job. It depends on which chemistry you're talking about. In biochemistry, we emphasize the liver is responsible for making sure your glucose levels stay steady in your blood, so they don't go up and down. So if you between meals and your glucose levels start going down, your liver will metabolize glycogen and it'll break it down to glucose and it'll export it. Okay, and if you eat a big meal and there's a bunch of glucose in your blood, then your liver will take up that glucose and it will turn it into glycogen. Okay, and this transporter plays an important role in that. This is a passive transporter. It says it right there. Okay, and from the cartoon, we don't see anything being hydrolyzed either, right? So based on just thermal energy, this transporter is doing this all the time. Okay, It's opening and closing and opening and closing. And if it's open to the outside and there's a glucose out there, it can bind, and then when it opens up the next time, it can come in. All right, same thing from the inside. All right, so what this does is this makes sure that your liver has the same amount of glucose inside as outside. Why do we need, so based on this, and it's a GLUT transporter, I keep saying glucose, I don't say monosaccharides, I don't say stuff. What kind of, what do we think this is a carrier or do we think it's a channel? And based on the cartoon. Carrier. It's specifically binding glucose. This is the one I talked about the other day. It'll bind glucose and not galactose. But it doesn't use any energy in making sure that only glucose can go across the membrane, and glucose is the same concentration out here as it is in here. And that, to me, is a really cool thing. Because what it means, we're not using any energy, and the liver needs to know how much glucose there is. It has other ways of signaling, right? We have insulin receptors. We'll talk about that as well. But if it needs to respond to how much glucose there is, it doesn't need to look outside the cell, right? I mean, it does some of that too, but that's the concentration in the cell as well. Okay, so that's pretty cool. So this is an example of a passive transporter that is not a channel, or not a channel. Any channel is going to be a passive transporter, right? Because a channel does not grab onto anything, and it can screen things sort of, but it doesn't grab anything and carry it through. Questions? I'm about to move on to carbohydrates. Okay? All right, so we're going to talk about carbohydrates now. So we're moving on from, we talked about lipids, and that had got us into membranes. And so we talked about transport across membranes for a while. But now we're finally at our final big molecule class, right? We talked about nucleic acids, we talked about proteins, we talked about lipids, and now we're going to talk about carbohydrates. And those are the big four if you think about macromolecules in biochemistry, biology. So carbohydrates are hydrates of carbon. It's right in the name, okay? So when we think about a carbohydrate, it's going to have a formula that looks something like that. It's going to be close to that. So you're going to have 1 water per carbon. And so let's say we're talking about glucose, okay? So glucose is a monosaccharide. It has the formula C6H12O6. I mean, you can see it right there, right? You guys see that? Okay. Now, we're going to chain these together, and whenever we link 2 of them together, what happens? I'm going to form a bond in biochemistry. What do? I lose a water, right? It's a condensation reaction. I lose a water. So if I put 2 of these together, it'll be that formula times 2 minus a water. Does that make sense? Every time you add them, you're going to have that happen. Now we're going to spend a lot of time talking about glucose because glucose is the most used molecule, but either straight out in biology or also modifications of it. And glucose is an aldose. So glucose is a 6-carbon aldose. And I have the structures of things here. So what I expect in terms of, there's some memorization in this chapter. I expect you to know the structure of glucose. And which other one do you want to know the structure of? I want you to, and it's the structure of something else so you don't think everything is glucose. Allose? I'll try to remember that, okay? allose. I don't even know what it does. But you need to know the structure of glucose and you need to know the structure of allose. Okay? And you need to be able to communicate these both in, this is a Fisher projection. All right, you probably remember those from organic chemistry. I'll talk about that in a second. And then also in a Haworth projection, and then also be able to do chair forms. Okay, and be able to interconvert between them. And I'm going to talk about how we do that sort of stuff right now. So first off, let's talk about Fisher projections. And essentially in terms of, before I go a little bit further, in terms of interconverting between these molecules or memorizing them, I don't know all of them. Actually, no, I actually have glucose and galactose memorized. But I can make, I only know glucose in its Haworth projection. And I just make the others from it. So I'm going to show you how we do that. But if we look at this, I'm going to actually abbreviate it. I'm going to stick form it. Okay. But if I look at glucose here, okay, glucose looks like that. All of them look like that. And then I can go ahead and draw 4 carbons across to show stereochemistry here. But what this really represents is that these carbons are coming out at us when we think about a Fisher projection in terms of stereochemistry. Did I say carbons? These bonds, right? Because for glucose, it's going to go OH, H, and I have to put my H's here because otherwise I think it's a carbon. Okay? And if these are coming out at me like this, right, these are tetrahedral, right, bonds, what's going on with this backbone? Is it how I drew it? Of course, it's not the way I asked it, but what should I be doing with the backbone? Mm-hmm. These are coming out, the other ones are going back, right? So really, when we picture these things as Fisher projections, it's a lazy way to draw them. It's not how the molecule is. The molecule is like curving back. It's curving around like this. Could be almost a ring. Okay, but so these are Fisher projections, and they are showing stereochemistry, because if we think about chiral carbons here, how many chiral carbons does glucose have? 4, right. All 4 of these are chiral. That only has 3 things attached, and this here has 2 hydrogens attached to it. So 2 the same 3. And that's going to be the case for all of these 6 carbon aldoses, which is why we get, so these are the D aldoses, and I'll show you the D depends on which way this one's facing, and then we have the L aldoses where we just take the mirror images of all these. Okay. So we end up with 16 of these, which shouldn't be surprising because there are 2 possibilities at each of these carbons. I have 4 carbons. 2 to the 4th is 16. Questions? All right. So you guys need to know allose and glucose. And glucose can also have, it can be in this linear shape, and it can also go to a cyclic shape. So let's talk about that. I'm going to have to go up and look at the structure of glucose if I'm going to start with the linear shape to show you this mechanism, because I only have it memorized in the circular shape, and I'm not going to drag you through my conversion technique just yet. Now I'm going to be lazy and not show them as wedges. But how can this get into a circular shape, which is what it does? Well, The mechanism for this, and we're going to do the mechanism, because what we're going to do is we're going to go from here to here. This is our product. What happens when I do a little squirrely line? It means it's either of the two conformations there, right? So the stereochemistry could be either one of them here. Okay, I'm doing that from memory, but I'll show you how you get to that maybe today. All right, so we're going to have this reaction. If I'm going to form a ring, I'm going to need a nucleophilic attack from this carbon. Not from this carbon, from this oxygen. All right, and so what I need is I need some sort of base around. Some sort of base will help it, right? We have a little bit of base in water, so you can imagine I'm going to grab that, and these electrons pop over here. And that's going to leave me with a negatively charged oxygen. What do we know about negatively charged oxygens? They're nucleophilic, right? Okay, and so I'm going to end up with a nucleophilic attack on this carbon right here. We're going to kick the electrons up. And now I'm going to draw this really funky. So I have this oxygen, and it has a bond that goes to there. Okay. And then I have this carbon. And this water up here, or this base, I'm sure it's a different water. I mean, so it is a different water because you're in water, right? But it's always elegant to come back and take that proton back from the thing you took it from, right? Okay, I'm going to count, make sure I didn't add any carbons. 1, 2, 3, 4, 5, 6. 6 carbons, great. That's what I start out with. When I grab that, then that's what it would look like if I changed this structure here after I picked up that proton to the Haworth projection. Questions? Shrink that just a bit so you can see all of it at the same time. And this is happening in solution all the time. All right, so we have a stereochemistry that is on these in terms of this nucleophilic attack. So you can end up with this nucleophilic attack is shown like that, but really it can come from above or it can come from below. So this is flat, right? Trigonal planar at this carbon. My attack can come from above or it can come from below, and depending on which way it comes from, I get a different stereochemistry on this carbon. This is called the anomeric carbon, and so we end up what we call two different anomers. So we have a beta anomer, which is where this goes up. And this is in equilibrium with the opened up form, with the aldehyde. okay? And that's in equilibrium with the circular form again, but in this case, we'll have the alpha anomer. Oops, that's galactose. Okay, does that make sense? So these things are in equilibrium all the time, and this is about 1%, and the other two add up to about 99% of, of the forms in solution. Questions? All right, so you see that we have, so I could ask you to draw me the beta anomer of, or the form of beta D-glucose. Okay, and that would be this is beta D-glucose. And this would be alpha D-glucose. Questions? I want to scoot really quick back. I want to show you the difference here between the D and the L's, how you know whether you're dealing with D or L's. These are D because they all do this. So on the farthest carbon that has a stereochemistry from the aldehyde, that makes this D. Okay? If I wanted to do, for these, if I wanted to do L-glucose, I would just essentially take the mirror image of what's going on for my glucose over there. And so I'm just going to go ahead and put my hydroxides on the opposite sides from where they are for D-glucose. Okay. But it would be, this is, oh, hold on. Let me not use that color. And I would know I was looking at the L version of the form of the sugar, because when I look at this carbon farthest from the aldehyde, it's on the left instead of the right. Does that make sense? How we know D and L? Okay. And then down here, our beta, our beta and our alpha anomers. Beta's up, alpha's down. Okay. The way I know this is D-glucose is because I think we're not going to go there, but with the L's of these. But if it was an L, this carbon would be down here in terms of stereochemistry. Okay, it would look really weird. They exist, they're just not natural, really. Questions before I move on? Okay, let me show you then. I've got this right here. So glucose gets used as glucose, but there's lots of examples where glucose gets modified. So you heard me mention some of these the other day when I was talking about your blood sugars. So there were some things I was naming that were forms of glucose that they had. So you have, here we have N-acetyl glucose right here. So at this position, there's an acetyl group, there's a nitrogen attached, there's an amine attached, and then it becomes an amide at that point. Here we have amines, so glucosamine. You may have heard of glucosamine before. Okay, there's just our glucose. There are all sorts of other groups, sorts of modifications that can occur to glucose, including adding, well, certainly phosphates. We see that in glycolysis. But we have some of these examples where we're actually even adding carboxylic acids. Okay? And we'll see those as we go through the various polymers that are made with glucose. Questions? Glucose and glucose analogs to make them. Okay, then let's go on and let's start talking about how these things come together to make polymers. And we're going to start off talking about polysaccharides. Polysaccharides are relatively linear forms of these polymers, and when we talk about polysaccharides, we're talking about generally a repeated unit. Okay, when we talk about oligosaccharides are going to be shorter. They're going to be maybe about 20, up to about 20 different sugars, and they're super highly branched. Okay, and they're used for communication. But we're not talking about those today outside of me mentioning those two classes. All right, so how do sugars come together? We could talk. What was that? Dehydration, right, exactly. Very good. Monosaccharides, let me make this big again. So monosaccharides join together through glycosidic bonds. Okay, well, that looks right to me then. So through glycosidic bonds. And so what you could imagine is if I had two glucose, right? So I've got one, this is going to be an alpha anomer of glucose. And I'm going to bring it up with another glucose. Oops. Okay, what I can do is I can eliminate this water, right? We're going to do dehydration, or I like to say condensation, and we're going to get our glycosidic bond between the two. Okay, so now I'm going to draw a weird bond again. And depending on where you go on the internet, they're not careful about this, but I think it's important to be careful about this, right? Whenever we go down and we cut an angle, right? If I was to do this, like you often see, what would I be indicating here? A carbon, right? There's no carbon there. So you will see things like that. So the best way to draw these is either have this come out, but it's going down to show me that it's still the alpha anomer, even in the joint state. Or what often gets done is you do this curved, okay, like that. And then it's going to come up and link to the other glucose, right? Still, it was glucose, so this OH is down, it's still down, so it's still glucose. And it's still the alpha anomer on the other side as well. Okay, and so we actually have a name for this. We can designate what that bond is. This is a 1,4, it's an alpha 1,4 glycosidic bond. All right, so what's happening is we're going from, if I think about these carbons, if we go all the way back up to our Fisher diagram, okay, let's not go to our Fisher diagram. Let's just do it here. Remember, this came from this. All right, anybody have really good memory? Were these on this side?
Yeah, I remember. Oh, yeah.
Okay, So we started with this, and the way we number these, we go 1, 2, 3, 4, 5, 6. All right, and so when we cyclize it, we end up with 1, 2, 3, 4, 5, 6. And over here we have 1, 2, 3, 4, 5, 6. And so this is it's a 1, 4. It's alpha because the 1 is going down, right? The one off the 1 carbon is coming down. It's alpha 1, 4 glycosidic bond. Does that make sense? Okay, so this is the bond you see. You see this bond is used in energy storage molecules. We'll say polymers. Okay, and so what I'm talking about there is I'm talking about glycogen. I'm talking about amylose. So glycogen would be the glycogen would be the animal storage polysaccharide. Amylose and amylopectin are plant storage polysaccharides. And this is the pectin you think about when you like add it to a jam or something to thicken it up. This is the same pectin. Okay, and that's different than what you see in structural polysaccharides. So structural polysaccharides use beta 1, 4 glycosidic bonds. All right, in terms of glucose molecules or modified glucose molecules. And so what that would look like if I was linking 2 glucose together, it's a beta, right? So it's going up. So there are 2 ways you could draw this. So you could do up. I do this sometimes. I didn't make that up. Oh, let's see, this next one's probably also beta. We're just linking 2 together here. Okay, that's one way to represent it. And another way it gets represented is you don't do the crazy S-shaped bond, but you flip these. Okay, so you do the first one, it goes up, so then the next one is upside down. Give me 1 minute. You can start packing and I will finish drawing it and just show it to you. You guys are so much more polite than my Gen Chem class. Okay, so this is going to go up, right? And then it comes down, but now my next glucose has to be upside down. Because it's glucose, and when you do a glucose, you're coming up from the bottom on this one, right? So this is the other way these get drawn.
Can you also draw it in like a staggered kind of, like with one higher than the other?
Yes, you could. So you're saying, could you do this?
Like
yeah.
That here.
Yes.
Yeah.
Okay. You guys have a good couple days. I will see you on Friday. No quiz.